Hello everyone. Thank you for joining the class today. Today we'll be solving Cambridge International AS and A Level Biology Paper 1 Multiple Choice, October-November 2021, question paper number 13. Question number 1. A student was asked to use the scale bar shown to calculate the magnification of a cell on a photomicrograph. Okay, so let's just assume that the distance between these 2 points are x millimeters. If we were to calculate the magnification of this, magnification is equals to x multiplied by 1000 divided by 2 would be the correct answer. Which method could the student use to calculate the magnification of the cell? A. Divide the diameter of the cell by the length of the scale bar with both measured in the same length of units. No. Measure the diameter of the cell in millimeters, multiply by 2000, and then divide the length of the scale bar measured in millimeters. Well, if we measure the diameter of the cell in millimeters, multiply with 2000 is a wrong statement. Measure the length of the scale bar in millimeters, correct. Convert it to micrometers, correct. And then divide by 2, perfect. So C is the correct answer. Which statement about light microscopy are always correct? The greater the resolution of light microscope, the greater the detail that can be seen. Yes, if the resolution is greater, then definitely we can see more details. Point number 2, the greater the magnification of light microscope, the greater the detail that can be seen. Well, we can increase the magnification as much as we want, but we won't see much details because there won't be resolution. Increasing the magnification of light microscope up to its limit of resolution allows more detail to be seen. Yes, that allows more detail to be seen. Shorter the wavelength of light used in light microscope, the greater the detail that can be seen. Correct. Yes, shorter the wavelength, greater the detail. So 1, 3, and 4 is the correct answer. Question 3. What is the length of typical prokaryotes such as the bacterium E. coli? The bacterium E. coli, so let's see, a typical E. coli bacteria can be like 1 micrometer or 2 micrometer in size. So 1.5, 10 to the power of 1, that means this is 15 nanometer. This is 150 nanometer. This is 15 micrometer. Sorry, this is 1.5 micrometer. And this is 15 micrometer. So definitely C is the correct answer. Question number 4. Which cell structures are present in Plasmodium? Plasmodium is a eukaryote, so ribosome, Golgi body, mitochondria, all will be present. 4A. Question number 5. Which cell structure may contain nucleic acid? Cytoplasm will contain nucleic acid, chloroplast, ribosome, all of this will contain nucleic acid except Golgi body. So 1, 2, and 4. B is the correct answer. Sorry, 1, 2, and 3. So A is the correct answer. Which row shows correct features of ATP? Carbohydrate in ATP is either a pentose or a ribose. Sites of production of ATP, mitochondria and chloroplast both can produce ATP. So B will be the correct answer. Question number 7 has been discounted from the syllabus due to its mistake that they have done in here. So we will move to question number 8. Which polysaccharide is correctly described? Amylopectin made of beta glucose, wrong. Amylose made of alpha glucose, correct. Storage molecule in plants, correct. Cellulose is not made of alpha glucose, glycogen is not made of beta glucose. So 8B is the correct answer. Question number 9, which formula represents a saturated fatty acid? A saturated fatty acid will have the highest number of hydrogen compared to that of carbon. So let's see. In all of these, there is 18 carbon. However, only A contains the most number of hydrogen. So 9A will be the correct answer. Question number 10. Which molecules always contain at least four double bonds? Four double bonds. Triglyceride molecule will contain three carbon oxygen double bond. So in the case of CO in the carboxylic acid. Okay. Collagen molecule, more than three carbon double bonds will be present. In protein, there will be always CO double bond NH present. So any molecule that has triglyceride will be cancelled. All right, triglyceride will be cancelled. So C will be the correct answer. Hemoglobin and collagen, because they have a lot of CONH bond. In the CONH, there is a CO double bond. So it will, so it will have, so it will have more than four double bonds. Question number 11. Which row about the structure of protein is correct? So to do questions like this one, it is good to actually attack it from the tertiary structure side. Okay, so let's see. The result of cross bonding between specific amino acid in the primary structure. All right. Cross bonding between specific amino acid in the primary structure. Well, we can't confirm that. The shape formed by folding of polypeptide and held together by hydrogen bonds. All right. So tertiary structure is held together by ionic hydrogen bonds, sulfide bridges between the R groups. The result of ionic and hydrogen bond disulfide bridges and hydrophobic interaction between the amino acids could be correct, formed as a result of interaction of side chains of amino acid in the primary structure. This is the most correct answer. All right. Secondary structure, left hand spiral formed by primary structure. The coiling of a chain of amino acid to form beta-bleated sheet. All right, coiling doesn't form beta-bleated sheet. Occurs because of attraction between hydrogen and oxygen atoms in the side chains. No, side chains must be cancelled. All right, formed by hydrogen bonding between amino acid forming the primary structure. Hydrogen bonding between the amino acid forming the primary structure. Correct. All right. For primary structure, the number of amino acid present in a protein. All right, primary structure is not the number of amino acid. All right, so for number D, the sequence of amino acid in the protein coded by mRNA molecule. This is the correct point. So question number 11, D is the correct answer. 12, which statement about collagen is correct? A collagen fiber is made of three parallel helices. Collagen fiber is made of many molecules of triple helices. All right, it is an insoluble fibrous protein with a quaternary structure. Yes, that is the correct statement. 1/3 of the amino acid making up collagen are valine. No. Collagen fibers are formed from several collagen molecules held together by ionic bond. No. So 12B is the correct answer. Question number 13. Which properties of water are the results of hydrogen bonding between water molecules? Solvent action. Well, hydrogen bonding does not ensure solvent action. So specific heat capacity, yes. Latent heat of vaporization, yes. 2 and 3, C is the correct option. Question number 14. Which description about all enzymes are correct? Number 1, catalyze the breakdown of large molecule into smaller molecules. All enzymes are not there to break down molecules. Some of them will build. Only function inside the cell? No. Enzymes can function outside the cell. Forms temporary bond with the substrate? Yes. Have a tertiary structure? Yes. 3 and 4 is the correct answer. So D, question 14, D is the correct answer. 15. The cells in the roots of beetroot plants contain a red pigment. When a piece of red tissue are soaked in cold water, some of the red pigment leaks out of the cells into the cold water. An experiment was carried out to investigate the effect of temperature on the loss of red pigment from the root cells. It was found that higher the temperature of the water, higher the rate of loss of red pigment from the root. The cell membrane becomes more permeable at higher temperature, and the protein tends to denature at higher temperature, which holds the cell membrane together. So which statement could explain this trend? Enzymes in the cell denature as the temperature increases, so the pigment can no longer be used for reaction inside the cell and diffuses out. Number one is a wrong statement. As the temperature increases, the tertiary structure of the protein molecule in the cell surface membrane changes, increasing permeability of the membrane. This is a correct point. Phospholipid molecules gain kinetic energy as the temperature increases, increasing the fluidity of the phospholipid bilayer and allowing pigment molecules to diffuse out more easily. Also correct. So when 2 and 3 is correct, that means B. 15, B is the correct answer. The diagram shows a partially plasmolysed plant cell. We can see solution X and we can see solution Y outside. What is found at Z? At Z, obviously, we're going to find solution Y. So C will be the correct answer. Question number 17. A single-celled organism lives in fresh water. Water that enters the cytoplasm of the cell by osmosis is collected into a structure called contractile vacuole. To remove the water, the contractile vacuole fuses with the cell surface membrane. All right. A student counted the number of times the contractile vacuole filled and emptied when the cell was placed in solution with different water potential. The results are shown in the table. The idea, well, here is very simple. If the cell is getting a lot more water, then there will be many contractile vacuole, and that will basically remove the water from the cell. So as the water potential is going to, as the external water potential is going to decrease, the rate of contractile unit vacuole emptying per minute should decrease. So that's what we see in the trend. All right. Which statement explain the pattern observed as the water potential of the external solution decreased? The water potential gradient between the cell and the solution increased, causing water to move into the cell more rapidly. No, water is not moving into the cell more rapidly. We can see that it is decreasing. The contractile unit is decreasing. All right. So the water potential gradient between the cell and the solution increased, causing water to move into the cell less rapidly. All right. And okay, less rapidly is correct. All right. And but then water potential between the cell and the solution does not increase. It doesn't increase. It decreases rather. Okay. The water potential gradient between the cell and the solution decreases, causing water to move into the cell more rapidly. Water is not moving into the cell, we can see from this particular table. So option D, the water potential gradient between the cell and the solution decrease, causing water to move into the cell less rapidly and the contractile vacuole to empty less frequently. This is the correct statement. So 17D is the correct answer. 18. Which row shows the correct number of each component of a single chromatid during anaphase of mitosis? Okay, correct number of chromosomes of a single chromatid. All right, during anaphase of mitosis. Centromere. Let's read one more time. Okay, question number 18. Which row shows the correct number of each component of a single chromatid during anaphase of mitosis? Centromeres, obviously centromeres will be 1. Polynucleotide strands will be 2. Telomeres will be 2. 18A is the correct answer. Question number 19. Some processes are listed: cytokinesis, differentiation, DNA replication, and mitosis. How many of these listed processes occur during tissue repair by stem cells? All right, cytokinesis occurs. All right, differentiation occurs, DNA replication occurs, and mitosis. All of the process 4. 19D is the correct answer. The photomicrograph shows the cell in various stages of cell cycle. We can see prophase. We can see telophase. We can see anaphase. And we can see cytokinesis. We can see interphase. This should be interphase. Which stage of mitosis? Which stage of mitosis is not shown? We cannot see metaphase. So metaphase will be the correct answer. Question number 20C. 21. Which letter is the key described uracil? All right. Is it a purine? No. Has a single ring structure? Yes. C is the correct answer. When a gene for protease is activated, which nucleic acid will be formed? All right, when a gene for protease is activated, so we activate a gene for producing a particular protein, which nucleic acid will be formed? So definitely mRNA will be formed. 22B. 23. The diagram shows a vascular bundle from the stem of a plant. We can see 1, 2, 3, 4. Which row describes the functions of the label cell? All right. Transports sucrose. Well, sucrose transporting should be done by 2. So 2 is phloem. All right. Transports mineral ions. 1 transports mineral ions. Stores starch. 3 stores starch. So B. Question number 23. B should be the correct answer. Some of the features present in the transport tissues of plant are listed. Okay, transport tissues of plants are listed. Number 1. Lignified walls. Lignified walls. Okay. Number 2. All right. We can see which features are present in sieve tube element. Sieve tube elements have cytoplasm. Sieve tube elements have mitochondria. Sieve tube elements have plasmodesmata. So 2, 3, 5 will be available. 24, C should be the correct answer. Question number 25. Which statements explain why transpiration is an inevitable consequence of gas exchange in plants? Number 1, hydrolysis reactions are taking place in cells. ATP production is occurring. Stomata must be open. Now the question, let's read the question again. Which statements explain why transpiration is an inevitable consequence of gas exchange in plants? All right, number 1, hydrolysis reactions are taking place in cells. No. ATP production is occurring. No. Stomata must be open. Stomata must be open. Number 3 only. 25D is the correct answer. 26. In an investigation, a leafy shoot was attached to a potometer exposed to a variety of conditions. The time taken for the meniscus to move 50 meters along the capillary tubing was recorded for each set of condition. Temperature, 20 degrees Celsius. Dry, humidity dry, air not moving. Time taken to move 50 millimeter, 125. Let's read the next question. Which row shows the fastest, slowest and fastest rate of water uptake for this investigation? Okay, technically at 20 degrees Celsius, when it is humid and the air is not moving, it takes the most amount of time to travel the 50 millimeter. All right, time taken to move 50 millimeter in terms of seconds. It takes the most amount of time. So the slowest should be 50 divided by 166. 50 divided by 166. 0.3. Slowest rate is 0.3 millimeter per second. And the fastest rate should be at highest temperature, dry, and all right, the least amount of time taken. So 40. The fastest rate should be 50 divided by 40, 1.25 or 3. So A is the correct answer. Question number 26, A. Question number 27. Proton pumps and co-transporters are used by plants when loading sucrose into phloem sieve tube element at a source. Which row is correct? First of all, proton pump. Proton pump pumps proton into a companion cell, raising its pH. No. Proton pumps pumps proton into a mesophyll cell, raising its pH. Pumps proton out of the companion cell into its cell wall. Pumps proton out of the mesophyll cells into its cell wall. All right. 27. Well, C is the correct answer. All right. Pumps proton out of the companion cell into its cell wall. And co-transporter, the high the concentration of proton outside, a companion cell drives the transport of sucrose into the cell. Yes. 27C is the correct answer. Question number 28. The statements list some of the events in the cardiac cycle. They are not in the correct order. Okay. The impulse travels to Purkinje tissue. A wave of excitation sweeps across atria. The atrioventricular node delays the impulse for a fraction of a second. The sinoatrial node contracts. The wave of excitation sweeps upward from the base of the ventricles. The ventricles contract, the atria contract. Which statement describes the sixth of this event to occur in the cardiac cycle? Okay, so we can see the sinoatrial node contracts. This should be the first event. After the sinoatrial node contracts, the wave of excitation sweeps the atria. This should be the second event. The atria contract should be the third event. All right. The atrioventricular nodes delays the impulse for a fraction of a second. This should be the fourth event. Fifth event, the impulse travels through Purkinje tissue. This should be the fifth event. And then the sixth event should be the wave of excitation sweeps upward from the base of the ventricles. So the sixth event is number 5. D is the correct answer. Question number 28, D. 29. Which tissues types are present in the walls of all blood vessels? Collagen, elastic, endothelial, smooth muscle. All right. All blood vessels don't have all of this. However, every one of them should have endothelial. So question number 29, D should be the correct answer. All blood vessels should have endothelial cell lining. Question number 30. The photomicrograph shows 3 white blood cells labeled XYZ. Which row correctly identifies these cells? First of all, cell X. Cell X is a neutrophil. So cell Y is a monocyte and cell Z is a lymphocyte. So D is the correct answer. Question number 30, D is the correct answer. When active tissues have high carbon dioxide concentration, oxyhemoglobin needs to release oxygen to the tissues. How is the carbon dioxide transported away by the blood from the tissue? As carboxyhemoglobin? Yes. As carbaminohemoglobin? Okay. As carboxyhemoglobin? No. Number 1 wrong. As carbaminohemoglobin? Yes. As hydrogen carbonate ions? Yes. So 2 and 3 only. C. 31C is the correct answer. As carboxyhemoglobin, only carbon monoxide is transported. Question number 32. Which effect does increasing carbon dioxide concentration have on hemoglobin? Hemoglobin is less efficient at taking up oxygen and less efficient at releasing oxygen. All right. Increasing carbon dioxide concentration, all right, does not, make it less efficient. It actually makes it more efficient. Hemoglobin is less efficient at taking up oxygen and more efficient at releasing oxygen. Hemoglobin will be more efficient in releasing oxygen. Hemoglobin is more efficient at taking up oxygen and less efficient at releasing oxygen. Question number 32. Which effect does increasing carbon dioxide concentration have on hemoglobin? You see, hemoglobin at high concentration of carbon dioxide will be less efficient in taking in oxygen, but more efficient in losing oxygen. So B should be the correct answer. Hemoglobin is less efficient at taking up oxygen and more efficient at releasing oxygen. Question number 33. The diagram shows a magnified section of part of the lungs containing specialized tissues. Okay, so part of the lungs containing specialized tissues, okay. Which row is correct for the structured label 1 to 6? All right. Contains high proportion of carbonic anhydrase. So we can see red blood cell is represented with 3. Red blood cell will have carbonic anhydrase, so 3. Hydrogen carbonate ion. Plasma will have hydrogen carbonate ion, so 5. 5 represents plasma. Lysosomes, all right. Uh, 6 represents a white blood cell. So lysosome will be contained within the white blood cell. So question number 33, C is the correct answer. Question 34. The table shows some parts of an animal's lungs that contains different cell types. Parts of lungs, trachea. Which parts of this animal lungs clean inhaled air and which carry out gas exchange? So gas exchange is carried out in alveoli, definitely. All right. So gas exchange is carried out in alveoli only. All right. And clean inhaled air, bronchiole, bronchus, trachea, all of them cleans inhaled air. So D is the correct answer. 34, D. 35. What helps to maintain a steep oxygen concentration gradient between the air in an alveolus and the blood? Breathing in brings in a supply of air with a relatively high concentration of oxygen to the alveolus. Correct. Blood flow brings blood with a relatively low concentration of oxygen to the alveolus. Yes, definitely. Deoxygenated blood enters the lungs. The relatively low concentration of carbon dioxide in the alveolus results in the carbon dioxide leaving the red blood cell, allowing hemoglobin to combine with oxygen. Perfect. So 1, 2, 3. A is the correct answer. Goblet cells are found in the trachea. Which cell structure would be extensively found in goblet cells? Goblet cells will have Golgi bodies, will have mitochondria, and will also have ribosomes. So 36A should be the correct answer. 37. In a country where malaria has successfully been eliminated, an outbreak of malaria can occur years later. What could allow the later outbreak of malaria to occur? Mosquitoes become resistant to insecticide. All right, mosquitoes, yes, mosquitoes can become resistant to insecticide. Migration of the population due to war, yes, this can bring in infected people. Those then can infect the mosquitoes and then people can get infected. Malarial parasites become resistant to quinine, yes. So 37A should be the correct answer. 38, which row about a person with leukemia only and about a person with measles only is correct? So leukemia only. White blood cells count in a person with leukemia increases. Bone marrow function in a person with leukemia only increases. White blood cells count in person with measles increases. So 38D is the correct answer. What is the cause of symptoms? What is the cause of symptoms of people with myasthenia gravis? Myasthenia gravis, antibodies block receptor molecules on the cell surface membrane of the muscle cells. Correct. Antibodies does block the receptors on the molecules in the cell surface membrane of the muscle cells. Question number 40. Tetanus is a bacterial infection. The graph shows the blood antibody concentration of two people. On day 0, person G was injected with antibodies to the tetanus toxin and person H was injected with vaccine for tetanus. What could be the result if G and H were infected with tetanus bacteria on day 20? Tetanus antibodies would not be produced in person G. If the person gets infected, the person will produce tetanus antibodies eventually. Antibody production would peak after day 32 in person G. Yes, because in the first event, it took around 12 days to produce the antibodies. Yes. B should be the correct answer. Antibody concentration would stay constant for person H? No. Second antibody peak would, in person H would be lower than the first peak. There is no confirmation on that. Could be higher. All right. So 40B should be the correct answer. Guys, thank you for joining the class. See you in the next class. Bye-bye.